So before introducing our speaker today for the Mark S. Brower lecture, I want to say a few words about Mark Brower. Many of you here in the room knew him quite well. He was a hematology oncology oncologist clinician, specializing in medical oncology. And this put him at the center of a complex, challenging diagnosis of treatment and protocols for hundreds of patients seen here at Weill Cornell and New York Presbyterian, including those with bleeding disorders, hematological malignancies, and breast cancer. Many of these patients are alive today because of his interventions, his guidance, and his ability to administer the latest advancements in treatment and medical protocols. He was beloved by his patients and also by his colleagues. It was an untimely death that he was killed by an auto accident at a relatively young age. And in memory of his his impact here and the love of his patients and colleagues, the Mark S. Brower MD Lecture in Hematology and Oncology was established through donations of former patients and fellow physicians. And this has allowed us to endow this lecture series. This will be the third Brower lecture. So a bit about our lecturer today, Sid Mukherjee. I've known Sid, I think I knew him a bit even when he was a medical student, certainly as a trainee at Harvard Medical School. So Sid got his BS at Stanford University, and there he was awarded a Rhodes Scholarship to go to Oxford, where he got a Ph.D. With a focus, I think, in biochemistry, or maybe he can correct that, but I think that's what it was, in immunology. He then came to Harvard Medical School as a medical to get an MD. And then stayed on at Mass General, where he did his residency and fellowship. Now, you can imagine, because I think there are many residents and interns here, that those are times in which you find yourself incredibly busy. But Sid, being a person of almost unbelievable energy, decided to write a book while doing his residency and internship. In this book, you will, for those of you who've seen it, you realize it's not just a small little book. It's actually a fairly large book. The title of the book was The Emperor of All Maladies, Biography of Cancer. And this book was so successful that he was awarded the Pulitzer Prize a year after it was published. And ultimately, it went on to become a series by Ken Burns in public television, PBS, which also was very well received. Sid has gone on to write additional books, the latest being The Gene, An Intimate History, which was just published this year. So, in addition, of course, to his notable literary career, Sid's real job, he actually has a job, is to treat patients and to do basic science and translational science to try to figure out new therapies. And his focus is primarily in the area of AML, where he and I are actually starting a collaboration now. So this is a really remarkable man who has had excellence in many different areas. Now on the faculty at Columbia, our sister institution with NYP. And it's a great honor to introduce Sid to give the lecture. Thank you.
So thank you so much. It's a real honor to be here. When Lou first asked me to give this talk, I just wrote a one-word email back to him. I said yes, because anything that Lou asks, I just say yes reflexively and then look at my schedule. So, but that's really a testament to the kind of scientist and leader that he is, and the way the institution has really been an astonishing place for him. I have one small anecdote about Cornell in particular, which is relevant, which is that when I was working on the Emperor of All Maladies, I wanted to see Papa Nicolau's microscope. Now, I don't know how many of you know that just a few floors above is Papa Nicolau's microscope, and you can actually call someone, I don't remember who it was, the librarian of the Papa Nicolau library, and if you should so desire, you can not only look at the microscope, you can actually look at the slides that Papa Nicolau made from the first series of patients to diagnose early cervical cancer. It's an astonishing thing, which is just 3 floors above you. So I was at Harvard then, I was at medical school then. So I took a train down, and I knew the institution, and went right up to this library, which is impossible to find. Basically, if you take 3 elevators and it's tucked in somewhere. How many of you have been to that place? 2 people. It's astonishing. 3. I would recommend doing so because, of course, that's one of the founding principles of the diagnosis of early cancer, pre-invasive cancer. It's really transformed our idea of how to take care of patients before their cancer has become invasive and Pap smear has saved the lives of hundreds of thousands, if not millions of women in the United States and around the world, certainly millions of women around the world. So an important institution. But what I cannot find, though, is a pointer. I knew you pointed it out to me? Yeah, thank you. Got it. So just a couple of thoughts before I start the talk. This is meant to be a very general talk. I will not talk about in great detail about much of the scientific work that we've started doing. We're a young laboratory, but we've had the opportunity to collaborate very widely, including with Lou's lab. I'm going to speak much more generally about what I think is a kind of landscape view of what is happening in the world of cancer. I want this to be a general talk. There'll certainly be enough scientific detail, I think, to carry out a further conversation if people want to do this. And for reasons you'll understand very soon, I call this personal medicine and the public good. Really, in personal medicine, I'll be talking about cancer, although some of these principles, I hope, and am confident will be actually applicable outside cancer to a much larger context of the question. So I wanted to start with an important slide, which I think hammers home a very essential point. This is mortality in the United States from all diseases, so all mortality in the United States. And as you can see, from the 1900s to 1990s, somewhere it stops around 1998, and you can see a general decline of mortality adjusted for various factors, including age. And a general decline, of course, is something you've seen in our own lifetimes being transformative. This peak, for many of you, of course, corresponds to the great influenza pandemic. But since then, there's been a general decline, which is very reassuring. That's that first curve. Here's what's more interesting and more complicated. If you look at the infectious component of that, so the contribution of infectious diseases to the decline in mortality, what you find is that this curve virtually completely mirrors this curve, whereas the non-infectious, the chronic diseases, looks more or less like a flat line. That's the challenge, actually, that we're facing, is that as the epidemiology of diseases transforms in this country from infectious to non-infectious or chronic causes, our contribution in all of medicine, in public health, to the decline of mortality from these non-infectious causes has actually been pretty minimal. So the question, meanwhile, of course, we have giant projects, including moonshot projects, personalized medicine projects, which, of course, hope to transform the management and treatment of chronic diseases. And the question we should ask ourselves is, okay, well, what have we done so far? What's the map of these chronic diseases been? In fact, the track record has been very, very sobering. In recent times, of course, we have seen a rise in one particular disease. I want to focus on that as a mechanism of contrast, and that is, as very many of you know, AIDS. This is the rise and subsequent decline of HIV/AIDS-related mortality in the United States, rising and declining in the 1990s. Most of that is a combination of public health campaigns and effective antiretroviral therapies, and you see this in greater detail over here, rising in the 1990s, AIDS deaths, then peaking in the mid-1990s, late 1990s, and then falling. And what's interesting about it is that there's been a slight dip in this curve as now some resistance has developed, and also the public health campaigns against HIV in some pockets of the city, or some pockets of some cities, have been declining. All of this is very important to remember, because I'm going to give you what I think is a brief history of this. So I'm just going to talk about this very generally, and I want to contrast this with some important themes in cancer. So a very brief history here. So early 1980s, we now know that the epidemic breaks out in New York City, San Francisco, and other places. In the early 1980s, it's tracked to a bloodborne or sexual transmission. The virus is identified. As I said, a vast public health campaign to prevent transmission is launched. Antiretrovirals are deployed, and resistance develops to antiretroviral therapies. Combination therapies are deployed in the 1990s, leading to, in many cases, a spectacular control of the virus. Antiretroviral therapies are partly personalized by the 2000s, and by that I mean that now there are mechanisms to test for resistance to certain viral, to certain antiretrovirals and viruses. Whereas, so it's partly personalized. At the same time, HIV becomes a global pandemic. Now, this is probably, in the United States, as I showed you before, one of the most recent powerful medical scientific crusades, followed by successes. But realize here that the personalization aspect of this, deploying antiretrovirals in a mechanism that is personal to an individual patient, really lies at the tail end of all of this. And if anything, has really become partly a late comer or late addition to the deployment of an effective campaign, both public health-wise and in terms of clinical medicine, against HIV/AIDS. It's an important lesson to remember, because in our lifetimes, we've only seen the arrival of personalized medicine for that sector quite late. So what about cancer then? Well, the history of cancer, which is covered in great depth, both in Emperor of All Maladies and in the Ken Burns documentary, can be told from ancient times. We know that cancer is a disease that's been present really in some of the oldest specimens that we have of human pathology. It's mentioned in some of the oldest books of medicine that exist. But really, to some extent, the modern era of cancer, the more contemporary understanding of cancer, begins with the definition of what may have been the first identification of cause. And this is Rudolf Virchow, who, as a young man, began to understand or realize, and again, people in this field might have the opportunity sometime to go to Berlin and look at Virchow's specimens, which are also publicly available for public view, much like Papa Nicolaus's are. But Virchow realized that, in fact, regardless of the tissue of origin, brain cancer, liver cancer, and other cancers, that fundamentally there was a process, a pathological process that united all of these cancers, and that was the abundant overgrowth of cells growing where they shouldn't be growing, cells growing in arenas where they shouldn't be growing, anatomically out of place, but also in manners, in physiological manners that did not seem to respect normal tissue boundaries. Virchow's second insight is perhaps even deeper. Virchow made the argument, an argument which has very deep relevance even today. Virchow made the argument that all cells must arise from other cells, omnis cellulae cellula, as he put it. And you could say, well, this is relatively obvious, but in Virchow's time, this was not obvious. There were all sorts of theories as to how cells arose. So Virchow made the argument that generally in all tissues, cells arose from prior cells. And if you take that argument at face value, and Virchow is obviously right in saying that, it turns out that it leads to a very profound realization about cancer, and that is that a cancer cell must necessarily have arisen from a normal cell. And if that's true, if a cancer cell must arise from a normal cell, then something must have happened to a normal cell to make it into a malignant cell. Some process, some black box process must have happened to transform a normal cell into a malignant cell. Now that idea, what that something was, is of course the idea that rings through much of this century of cancer medicine, cancer biology, and cancer research. What is that something that allows a perfectly good citizen normal cell to be transformed into an abnormal cell, which divides and proliferates abnormally, has abnormal metabolism, has abnormal characteristics of growth, invades tissue, and ultimately causes death? Keep that thought in mind, because we'll come back to that over and over again. In the meantime, without the knowledge of what that abnormality was, in the absence of the knowledge of what the cause was, the first attempts were made to extricate or extirpate cancers without addressing their cause. In other words, if you could cut the cancer out, you could then have possibly removed the cancer in its entirety. And as we said before, the closer you got to the cell of origin, the site of origin, the earlier you got to the intervention, the more successful the chances of that therapy. This led very soon after Virchow's observations, independently really, to the golden age of cancer surgery. This is a picture from a clinic in Vienna with Bill Roth, one of the great cancer surgeons, performing among the first extirpations of cancer, the removal of cancer from its organ, in this case abdominal cancers with Bill Roth attack. And Bill Roth's student, as many of you know briefly, but one of Bill Roth's students was William Stewart Halsted. Halsted, again, as many of you might remember or know, was really a champion of cancer surgery. He was one of the great cancer surgeons, practiced in New York and then moved to Baltimore, where he launched what he would call, and others would call, a radical attack on cancer. Halsted believed, importantly for his time, Halsted believed that the reason that patients were not being surgically cured of breast cancer, this was the cancer that he was most interested in, was because they were not being treated aggressively enough. The cancer was not being removed aggressively enough, and Halsted and his students therefore launched increasingly aggressive surgical attacks on cancer, leading to the ultra-radical, the ultra-radical, and the super-radical mastectomies, in an attempt, as he put it, to completely remove the tumor and thereby rid the patient of the cancer. The hypothesis was a powerful hypothesis. It seized the imagination of a nation and a world increasingly obsessed with cancer. But sadly, the hypothesis was not completely correct. And it was not correct because, for reasons that we understand now, and that is that for women in whom the cancer had already spread beyond the breast, in other words, for women who had metastatic cancer to begin with, the more aggressive removal of local cancer makes no difference. The cancer is already out of the local site. And ironically, for women in whom the cancer has not spread outside the local site, more aggressive surgery is simply more aggressive without giving them the benefit of local surgery. So in fact, it's a perfect mismatch for both the circumstances in which a patient might present with cancer. The problem was that it was impossible to run that trial to prove this point, partly because of the culture of medicine. We'll come back to this idea over and over again, but the culture of medicine was inherently resistant to testing a hypothesis that seemed very true at face value. And in fact, patients themselves would rather never be randomized to the non-radical arm of the surgery. They didn't want the less radical surgical operation, and it was impossible to run the trial. Ultimately, it took about 100 years after Halsted had launched the mastectomy to run the first trials comparing radical mastectomy with local mastectomy plus radiation, and that trial was negative. It was so surprising a result that it was repeated at 10 years and at 20 years, and it still remained negative and has become one of the cornerstones. Now, why is this relevant? It's relevant because even from this early understanding, it became quite clear that an individual cancer had to be treated on a stage-by-stage basis. In other words, deeper information about an individual cancer spread, in this case, outside or within the localized confines of the breast, was relevant in launching the appropriate treatment regimen. So right from the very start, even with the surgical history of cancer, there was an attempt early on to try to stage patients. And what is staging, aside from a very, very crude mechanism of personalizing cancer therapy? So in fact, what we call staging today is really step one of personalized cancer therapy, because it is an attempt to try to understand where the patient sits on the spectrum of early disease, intermediate disease, maybe late metastatic disease, to try to direct appropriate therapy for those patients. Well, so if surgery was trying to reach its own, make its own peace with staged disease, appropriate staging of cancer, there was also in medicine, in medical oncology also, the same kind of realization was slowly dawning. We've turned the clock back a little bit. As early as the 1900s, Paul Ehrlich, the chemist, had made the argument that aside from the surgical extirpation of cancer, there could be medicines that would specifically kill cancer cells and therefore be systemic medicines. They would go through the whole body, and the chemicals would now kill cancer cells as opposed to removing the cancer cell. And as many of you know, one of the first chemicals to achieve this was they were borrowed from the world of chemical poisons. Mustard gas at Bari was noted to be a potent toxin for hematopoietic cells, in particular lymphoid and to some extent myeloid cells. And very soon, these kinds of gases, mustard gases and other poisons, chemical poisons, were used in smaller doses to treat patients and send them into remissions of cancer. So the early history of chemotherapy really begins with the deployment of empirical poisons, poisons found empirically from war gases, from other toxins, and the use of those toxins to try and kill cancer cells to try to get sustained remissions and potential cures. Again, why is this important? It's important because if we go back to Virchow's idea of transformation, recall that none of these therapies, none of these empirical therapies have really any dependence on an understanding of that process of transformation. There's no understanding built into any of these chemicals about what causes the movement or the transformation of a normal cell into a malignant cell. Most of these were inhibitors of growth, and so what they attack is the phenotype of cancer. They attack what happens as a result of the consequence of transformation. They are not in any way directed towards understanding or treating the causal mechanisms of carcinogenesis, at least with these poisonous chemicals. Nonetheless, they were very successful in instances, and particularly successful in combination therapy. So at the National Cancer Institute, Emil Frey and Emil Freyreich in the 1950s were able to now combine not one, but several of these empirically found chemical poisons, often in combination, often with deep toxicities, in this case and in their case with childhood lymphoblastic leukemia, ALL, and were able to lengthen the remissions and achieve, in fact, complete cures of patients with childhood ALL with empirically derived combination chemotherapies. And similar principles were applied to Hodgkin's disease. This is Henry Kaplan at the NCI and then subsequently at Stanford, now combining radiation therapy with some of these chemical toxins to get sustained cures for late-stage Hodgkin's disease as well. Similar principles were also applied to breast cancer, although with less effect or less success. Nonetheless, this led to a wellspring of interest in the usage of combination chemotherapies combined with surgery and radiation to potentially cure all cancers, leading to, in 1969, this public advertisement from what would eventually be called the Laskerites, the Citizens Committee for the Conquest of Cancer, which says, Mr. Nixon, you can cure cancer. Notice a single C cure, single C cancer. One cancer, one cure, somehow or the other, like locks and keys, combinations of chemical therapies used in concert with surgery and radiation would now cause all cancers to yield in some process. And if you read the fine text, this is printed in 1969, if you read the fine text, the outermost edge of that was 10 years. So by 1979, this advertisement would have you believe that all cancers would be completely conquered using this kind of strategy. Well, of course, that did not happen. This is the rise in the National Cancer Institute budget during this period of time, going up from just a smattering of $1 million to several $1 billion, as you know, over time. And this is the flattening in blue for men and for women, the flattening of cancer-related mortality, but not that rapid decline that had been seen for infectious diseases that I showed you in front, or for that matter, for not even for men and women, again, a rather modest change. The red curve shows the expected rise in incidence. And most of this, as many of you might know, was because of a decrease in lung and bronchus cancer from smoking cessation. So anti-smoking public health campaigns drove down the mortality from lung cancer and bronchus cancer. So much of this flattening is related to this, although certainly other things have contributed, including a decline in cancers of the prostate, cancers of leukemias, which are driven by new treatment paradigms, and other cancers, including breast cancer. So by the mid-1970s, by the early 1980s, the question had returned, to some extent, the field had returned back to the drawing board to ask the question, what should happen next in terms of our understanding of cancer, and how would that understanding be used to motivate a new series of attacks on cancer, whether they be towards prevention or treatment or cure? Well, the question of what the phenomenon of cancer was caused by, how cancer became, how that transformation occurred, as Virchow had pointed out, went back to observations that were made really throughout the early 20th century and even before the 20th century. It was well known that heredity contributed to cancer. This is a picture of Hilario de Juva, who was a Brazilian ophthalmologist who noted that cancer ran in families. In fact, it would turn out later that this was a very, very important genetic link. We'll talk about that later, but the Brazilian ophthalmologist noticed that there was clearly a hereditary predisposition to cancer. Several other hereditary predisposition syndromes to cancer were subsequently identified, clearly implicating that genes were somehow related to cancer, although it was not clear why. Long before these families had been identified, Percival Pott in England had noted that chimney sweeps had a striking incidence of scrotal cancer. And Percival Pott made the observation that this must be because of an environmental substance, which he in fact called a carcinogen, that was causing this increase in cancer, particularly in that population of exposed scrotal chimney sweeps. There was long known that there was an element of chance, and of course down the road here at the Rockefeller, Peyton Rouse identified viruses that were linked to cancer. So somehow or the other, whatever the cause of cancer was, had to integrate theories of heredity, environment, chance, and viruses. And that moment really came in the 1980s, late 1970s into the 1980s, when the hypothesis was made that mutations in growth-controlling genes, subsequently other genes that are also control growth, maybe through secondary processes, but mutations in growth-controlling genes cause cancer. There were many actors in this. I've identified some people: Harold Varmus, who's now at Cornell, Bert Vogelstein, Bob Weinberg. There's a whole list of names here, many prizes, many important awards for this process. Harold himself put it very nicely, borrowing from Beowulf, thereby describing cancer as a distorted version of our normal selves, referring to the idea that basically the very same genes that play a vital role in the normal physiology of cells, the genes that control embryogenesis, the genes that might control the way cells respond to each other, how they stop growing when they contact each other, the way cells respond to their environments, etc. The normal physiology is distorted. Mutations when mutations occur, you ultimately get a cell that it does not know how to stop growing, does not respond to normal signals, and acquires autonomous growth, thereby unifying much of this. And this, of course, explained how all of these different phenomena could cause cancer as its center. The mutations could be inherited. In the case of familial cancer syndromes, they would come from your parents. In the case of the environment would create these mutations. You could acquire the mutations by chance. When cells divide, there's a natural error in copying genes. And certainly viruses could also introduce these mutations exogenously into cells. So all of a sudden, the mystery of multiple causes converged onto the idea that mutations in growth-controlling genes cause cancer. I won't spend a lot of time describing these mutations or describing these genes. Most of them are familiar to people who work in cancer in this audience. So the question then was, okay, so if alterations in growth-controlling genes cause cancer, how many are they? How common are they? Is there one gene? Is there one gene per cancer? Is it one gene for breast cancer, one gene for leukemia, one gene for lung cancer, et cetera? Or what is the spectrum of mutations? Again, for people in this audience who are now living in the post-genomic era, this seems like an obvious question, but in the 1980s, this was a very important question, and there were lots of differences of opinions. People thought breast cancer had a master gene that was mutated, lung cancer had a master gene that was mutated, or maybe they were common to all cancers. Many people, including most importantly Bert Vogelstein's group and other groups, showed that there was not one gene but many genes. Even in a single cancer, multiple genes needed to be mutated. In fact, a cell did not suddenly wake up one morning and undergo the process of transformation that Virchow had described. It in fact slouched towards transformation. Multiple mutations were accumulated in cells. In this case, a normal cell slowly transforming into a carcinoma through a series of mutations in well-defined events in genes. So that therefore begs the question, if there are many genes, then now the question is, what combinations are present in individual cancers or in individual cells of a cancer? Is everyone with me so far? So we're now at a moment where we're beginning to now look beyond individual genes in cancer towards genomes of cancers. And that had to wait for the technology to catch up. That technology caught up. The capacity to sequence not one gene, but multiple genes in a single cancer specimen eventually caught up in the 1990s and towards the 2000s, leading to a real genetic picture of cancer. And we're going to pause for a moment to try to describe that as simply as possible. So here is one sort of schematic of this. So if you imagine the human genome stretching from its very first gene, like a snake, on a blank sheet of paper, and the very last gene is on this corner. And you have a patient, let's say with breast cancer, who now comes to the clinic, and you have the capacity, as we now do, to look at the entire genome, or perhaps at the what's called the exome of these patients, so the active parts of the genome, and mark with a dot every single genetic mutation that occurs. So this is just a schematic or an abstraction of the actual kind of information that you get. And you mark, in this one woman with breast cancer, you mark 1, 2, 3, 4, 5, 6, 7, 8 genes that are mutated in her breast cancer compared to her the baseline genome that she has, the normal genome that she inherits. Now imagine that her identical twin also develops breast cancer. She develops breast cancer maybe at the same period of time. The lesions look exactly the same under the microscope, so they're both classified in similar ways pathologically. And yet, when you look at her genetic fingerprint of the identical-looking breast cancer, you find that the genetic fingerprint is similar, perhaps, but is different in a couple of genes. So now you have basically, in this particular patient with similar-looking breast cancer, the fingerprint, the genetic fingerprint is different. And for another patient also with breast cancer, the genetic fingerprint might be yet different. So here is an important conundrum, which I think begins to outline some of the problems, one of the problems that we think about in cancer, and that is that every individual specimen of cancer is its own disease, or every individual specimen of cancer is its own specimen of cancer. And the challenge, I think, for cancer biology is to find a paradigm in another disease where every single example of the disease is its own example of a disease. That is where one of the challenges, not the only challenge, but that is one of the challenges in cancer, is where the n of the number of patients equals n of the number of variants of disease. And that challenge has now made itself very apparent as we sequence cancer genomes even more deeply. What you can do, however, is you can begin to organize this information. So one way to organize this information is to take multiple specimens of, let's say, breast cancer alone, and make big peaks out of common mutations, small peaks out of uncommon mutations, and obviously less small peaks out of the least common mutations, and you get a map that looks like this. This is work done by Bert Vogelstein, but again, others have replicated this work. I'll show you some of our own work in leukemia. So it looks like this. This is a kind of a schematic map of, in this case, colorectal cancer, which with big peaks corresponding to very commonly mutated genes, small peaks corresponding to less commonly mutated genes, and single dots. So this one dot would be mutated in one particular specimen of breast cancer, colorectal cancer, sorry, but not in other specimens of colorectal cancer. So everyone with me so far? So this, of course, captures the level of diversity, but also combines it sort of into a single map of in this case, one particular cancer, such as colorectal cancer. And this just points out that level of diversity. So what does that mean in terms of, this is our own data, what does that mean in terms of individual patients? Here's a much more limited panel. I think there are 1, 2, 3, 4, 5, 6, maybe 8 or 9 genes here. So one patient, this is patient 1, has a mutation in this gene, and this gene. So this is one patient. A patient 2, interestingly, has exactly the opposite. So where there are holes here, there are mutations here. And yet, when you look at them, in this case, we're looking at them in a clinical trial, both seem to cluster in response in one direction, whereas other patients don't cluster in another direction. So what has become, in this case, it's become a kind of mix and match problem of gigantic proportions, where you're trying to find the mutational spectrum that a patient has and match it to therapy on one hand, and trying to figure out how to make these matches in a way that are sensible, that make sense in terms of biological pathways and other parameters of the cancer. So has that approach worked? It's worked very well in circumstances where the mutation is very well defined and you happen to have a powerful inhibitor against that particular gene that's driving the physiology of the cancer. So here's probably the best known example of that, and that's from a disease called chronic myelogenous leukemia. I've used the same schematic plot from the colorectal cancer, but of course this would be unique to chronic myelogenous leukemia. You'd have different genes that are peaks and troughs. In this case, a particular genetic mutation, a fusion between two genes, was found to be very, very common across virtually all cases of chronic myelogenous leukemia. And an inhibitor, a chemical inhibitor, was synthesized that would sit in the cleft of the protein made by this mutated gene. This is a drug called imatinib or Gleevec. This is amazingly, it's a chemical entirely synthesized by humans. It's an astonishing thing. So this is a chemical made from scratch by humans to lodge into this cleft. It was discovered through a screening process, but found to lodge into the cleft of the protein made by this mutation. And many of you know that this led to one of the most successful treatment paradigms for this particular disease, chronic myelogenous leukemia. Here's the pivotal clinical trial comparing chemotherapy. This is a Kaplan-Meier plot looking at death or relapse, actually survival in this case, with traditional chemotherapy, comparing with an old therapy, interferon, versus Gleevec, where you really don't see that. So you see basically prolonged survival. And this is a very, very significant result in cancer. So by the 1990s and 2000s, actually by the 2000s I should say, the idea that we could find Gleevecs for every cancer became a very tantalizing idea, that you would genetically profile the cancer, find the driving mutation, a common driving mutation or a common driving genetic mechanism for that behind that cancer, and you would be able to find an inhibitor of some sort that would turn it off like a lock and key. Was a dominant paradigm, and it was embraced very successfully by the world of cancer, ultimately being known with various names, included targeted therapy, was a very, very dominant paradigm. And this was repeated somewhat in other cancers, although I would say, in all fairness, not with the spectacular success of chronic myelogenous leukemia. This is again, a schematic of breast cancer, and the HER2 gene, usually in amplification. We, as a community of scientists, made from scratch a molecule that binds and inhibits or kills, these cancer cells. This is a picture of Barbara Bradfield. She was patient 0 to take Herceptin. She was basically the first patient to receive Herceptin, and is still alive today. She had metastatic HER2 positive breast cancer, was treated with Taxol and Herceptin. And in fact, this is my picture. I contacted her when I wrote the book, and I sent her a package of Herceptin. And her husband took her picture in her backyard. She's still alive today, actually, being the first person to enroll in the clinical trial in California for HER2-positive breast cancer. And here's the result of the initial trial of Herceptin in breast cancer, again showing very clearly in the Herceptin-treated group versus the non-treated group a clear benefit in survival. In the initial trial, unfortunately, many patients would develop resistance and relapse. But what was astonishing, and Lou and I were talking about this, we now know that if you treat patients in an early stage, if you remove a HER2-positive tumor in an early stage and give Herceptin in what's called an adjuvant setting, to basically, you can imagine, to remove any microscopic or residual cancer, exactly the paradigm that had been used in the past for other forms of chemotherapy, we now know that has a remarkable effect. So in many cases, they achieve very, very long-term relapse-free survival. So this has really been a paradigm shift in HER2-positive breast cancer. The problem, of course, was that this approach of somewhat, genetically annotating based on individual genetic mutations or gene signals or markers in individual cancer cells was very successful when you could have a medicine against that. Gleevec is a great example of that. Herceptin is a great example of something that marks those particular tumor cells, and in that case, an antibody is very helpful. But the question, very often, it was very hard to find such inhibitors, such exquisitely targeted inhibitors of these genes. So one question that was raised at this time, had been actually raised long before, is rather than inhibiting these individual genes where you could inhibit them, is it possible to inhibit the network or pathways of communication that exists? So obviously for a given cancer cell, to start acquiring its malignant physiology, the mutated genes in that cancer cells may communicate with each other, forming pathways. And these pathways may also be targets for various drug therapies. In other words, the pathway would be activated in cancer cells for some reason, be less active in normal cells, and if you could somehow modulate the increased activity in the cancer cell, sparing the activity in the normal cell, then you would have an anti-cancer therapy. It would also be somewhat personalized in the sense that it would be attacking a pathway that was unique to that one particular cancer cell. So you can imagine, you can create an analogy here to the parts of a very complex machine. So rather than, of course, capturing the mutations in individual cancers, that you would be attacking, in this case, the physiology of cancer, metabolism, growth, immune escape, etc. And just to point that out, you can imagine the cancer cell is a complex machine that ultimately has disrupted metabolism, disrupted growth, that you would attack some part of it that controlled some aspect of the machine that controlled one feature versus the other. So for instance, the aspect that might control its immune escape, the capacity of the cell to escape the immune system and grow, or its malignant growth potential, or its abnormal metabolic usage, etc. And this understanding converged in the 1990s with the idea that, in fact, when you looked at these pathways that are abnormal in cancer cells, you could organize them into some hallmarks. This diagram keeps changing, but just to give you some sense of it, evading immune destruction is a common hallmark that exists across multiple cancers. The instability of the genome and the acquisition of mutations, what's called replicative immortality, the capacity of the cell to grow over cycles and cycles and cycles, inflammation, the capacity of a cell to pull out blood vessels and so forth. I won't dwell on the hallmarks, but again, the idea that cancers, regardless of where they exist or how they exist, that they may have some common features or commonalities, and in fact, attacking some of these might be relevant or responsible for again, in new generations of medicines. And again, to liken that to this complex machine, the idea would be somehow to jam up this cog or cut up this wheel and thereby arrest the development or arrest the progression of the cancer or the movement of the cancer itself. So That idea was tested, or was put to test in a trial, which I want to focus on sort of in the last part of my talk, because it's a very important trial. So that's called the Shiva trial. How many people have heard of this Shiva trial? So it's an important trial, mainly because it was negative, but I'll go through it. It was done mostly in France, 741 patients. They underwent molecular profiling of individual cancers, so their individual cancers was molecularly profiled. And then they were given targeted therapy based on a physician's choice. So basically, 3 pathways, if you can imagine 3 of these pathways were identified. 11 drugs could be chosen based on the 3 pathways. So you got nominated into one of 3 pathways. You could have 11 drugs that were chosen. So this was attempt number 1, as it were, to deliver personalized medicine based on molecular analysis of individual cancer patients. And the result, unfortunately, was negative. So this was a multi-center trial. It was a phase 2 trial. And basically, there was no benefit for the personalized arm versus the non-personalized arm of this trial. This brought a huge set of critiques within the world of cancer. As I said, it was an early trial, but it brought a huge set of critiques. And indeed, if you looked at the individual pathways that were analyzed, PI3 kinase inhibition, hormone receptor, and MEK and ERK, none of these 3 individual pathways was personalized medicine in this context successful in changing the survival or progression-free survival of patients. So this led to a nice editorial, which is called Sitting Shiva on the Shiva trial. But more importantly, the question that was raised is a very important question, which is, why did this trial fail? Of course, it's an early trial. It was, it's really an early study, phase 2 study. Why did this trial was not successful? Well, one possibility, and so remember, it is successful in individual cancers. So if you take breast cancer and you annotate it for estrogen receptor positivity versus negativity, there's clearly a signal that those patients will respond to, for instance, tamoxifen. If you take leukemia, which is BCR-ABL positive, it's very clear that those patients will respond to Gleevec. And yet, if you now forget the tissue of origin and you take patients and you molecularly profile them based on pathway analysis, you've suddenly lost some information such that now your molecular annotation is no longer successful. So why? One possibility is that patients develop resistance. The second possibility is that the drugs were not potent enough. We don't know you don't have the right drugs for PI3 kinase, for instance. We were giving the wrong drugs. They were developing resistance for other reasons. Or that the pathways are unknown, unimportant, or as I said, are tissue context specific. So they're specific. So as I said, to remind us again, tamoxifen works spectacularly in ER-positive breast cancer, but somehow or the other, when you lose that context, you now suddenly have a situation where, at least in this early trial, it didn't work. So then the question is, what do we do if that's the case? So do we go back to combination therapies? Do we go back to immune therapies? Do we create more potent pathway inhibitors? Do we empirically test cancer cells for their capacity to respond or not respond? Or do we now return back to primary and secondary prevention? And to some extent, this is where the field of, in my estimation, this is where some of the most interesting questions in cancer therapy are sitting now. Should we deploy combination therapies after molecularly targeting or profiling cancers? Should we use immune therapies, which may or may not care about what the mutations are, but will kill cancer cells based on some other characteristics? Should we create more potent pathway inhibitors? Are we really using the wrong drugs? Should we forget all of this? Maybe we don't know any of this. Should we be testing individual cancer patients' cancer cells? Or should we focus on primary and secondary prevention as a backup? Of course, the answer is we're doing all of these in parallel. None of them are exclusive with each other. In fact, they intersect and interact with each other. And in the last few minutes, I want to focus a little bit on these sort of 5 paradigms. Well, one paradigm that combinations of targeted therapies can work comes from the multiple myeloma world. And it's a very powerful paradigm. So in fact, if this is an aggregate data from multiple myeloma, about survival, the proportion of survival, if you were diagnosed between 2001 to 2005 versus if you were diagnosed between 2006 to 2010. In fact, I've seen more recent update of this, and if you were diagnosed from 2015 onwards, it seems to be, it's really early days, 2010 onwards, it's really early days, but really the curve flattens out even more. So what happened in multiple myeloma? Well, one of the things that happened in multiple myeloma is that it was one of the first cancers where combinations of targeted therapies were effectively deployed. There are a variety of reasons for this. We can talk about that afterwards. But really what happened is, individual parts of the multiple myeloma, individual part of it was the individuals with multiple myeloma contributed very, very powerfully to clinical trials. There was an active consortium, and that continues even today and has changed the landscape of survival in multiple myeloma. It's one disease where really we've seen in our lifetime a dramatic change in the survival largely based on targeted therapies, 2, maybe 3 important targeted therapies, and now even new ones coming online. The second paradigm is using immune therapy. Now, the interesting thing about immune therapies is, of course, that these immune therapies are largely, not completely, but largely agnostic to the mutations inside cells. They recognize cancer cells by virtue of abnormal characteristics of the cancer cells that maybe reside on the cell surface of the cancer cell. Again, abnormal physiology, rather than particularly caring about what the mutation is. That's not absolutely true, but it's proven to be largely correct for in many cases. So there are 2 variations of flavors of it. Either you can engineer T cells and re-inject them. This is kind of a gene therapy paradigm in which you remove a T cells from a human being's body, engineer them to now become activated against the cancer and re-inject them into the individual. One of the successes of that has been the so-called CAR T cells that have been re-engineered to become, as Michel Sadelain has called them, living drugs. These are cells that are re-injected into a human being, and this is actually one of patient zeros, not the only. There are several patients in the first cohort. Emma Whitehead, who still remains, actually she's profiled in the film, still remains in a really spectacular remission from these re-engineered cells that have been re-injected back into her. She had tremendous relapsed acute lymphoblastic leukemia, really recalcitrant to all therapies, including transplantation. And this is some data from that. I'll skip over that for a second. And the other version of that is to not engineer T cells outside the body, but to re-educate the T cells to attack the tumors within the body, as it were, to resensitize them or to reactivate them so that the cells can go and trigger and kill cancer cells. And that's based on the idea that when a tumor cells and T cells have a complex interaction, and sometimes when they coexist, the tumor cell can essentially create signals that inactivate or make the T cell or the immune cells insensitive to being detected by the T cells. So in other words, it's like a two-handed handshake, which allows these cells to become invisible to the immune system. This interaction can be disrupted, as we now know, by various medicines, and that has led to one of the things that I had never suspected would happen in my lifetime. I had made a bet with someone that during my lifetime we would never see a successful therapy for smoking-related lung cancer that made any difference. In fact, I lost that bet, thankfully, because in the pivotal trial, this is of one of these mechanisms to disrupt the interaction between the cancer cell and the T cell. There's a survival benefit in lung cancer. In fact, it remains true for smoking-related lung cancers. And in fact, this line flattens out and remains flat. Actually, the last I heard at the last ASCO meeting, remained flat at least now for another 1 year, which means that there is a hint that these patients actually have real benefits that persist over long periods of time when they're treated with these so-called checkpoint inhibitors. What's interesting about this, that even these inhibitors are now getting a personalized aspect to them. So patients where you can actually see that these pathways of inhibition, this kind of invisibility, the cloaks of invisibility, are particularly active, can be identified as a biomarker. And what's interesting, just last week there was a paper showing that in fact you can track the resistance, again, to a genetic pathway that's unique to certain cancer cells and not to others. So you can ask the question up front and as the patient is being treated, who is responding, who's not responding to the therapy based on the individual specimens of cancer. And then the final one that I wanted to mention is that, occasionally, this is actually from our own clinic, and I talked a little bit about this in an article in the New York Times. In our own clinic, we have started to, where we cannot find genetic markers in cancers that are predictive, we've started now looking at large panels of drugs and asking the question, can we empirically decide or define which drugs are active against cancer? We extract the cancers from the bone marrow in this case, test them against about 750 drugs that have all been FDA approved at 4 doses, and ask the question, which ones are active or inactive. This is how that process works. I'll spare you the details. We've tested about 10 patients thus far, and once in a while, with a couple of patients, you can define new or previously unannotated or unidentified drugs that may be active, and then we have to requisition individual patients. We have to figure out how to pay for them. We have to requisition individual patients to figure out how they're being treated on these individual drugs and how to mark their individual therapies. It's a much more complicated process than you might anticipate. So that really brings us back to the central question, which is public good, which is the question that I'd asked, and that is that really, can we imagine a paradigm in which we can use personalized medicine at this scale, at an individual level scale, to drive down the mortality from cancer at least, and what the challenges in that case are? This is something, historically speaking, it's a paradigm that has never occurred at least in the history of medicine. And the challenges remain extremely steep. And therefore, the questions of how to coordinate the prevention, detection, and cure of cancer remains even greater today as we personalize cancer medicine. So, in sort of coming to a conclusion, we now know that cancer physiology involves several hallmarks of cancer. We think that combining hallmark therapies in a personalized way is likely to yield vast successes in this next phase. But then it is to be balanced against the larger question that historically this has not been an approach we've ever used with success with really any other disease in the past. Few exceptions aside, I talked about one of them at least in the past. So what's happened as a result of that is that we're shifting paradigms, although, as I said, we're shifting them with a certain degree of humility and a certain degree of trepidation about what happens next. Of course, there's an attempt to, as I showed you before, find responders versus non-responders and then move them to trial, as opposed to the old method in which you did the trial and then figured out who was responding versus non-responding. This will lead to a deeper understanding of the science and hopefully reduce the cost of failed medicines, because the medicines will be appropriately directed to the appropriate patients. But the major themes that remain is, how do we counter the costs? How on earth are we going to figure out how to deliver medicine at this scale? If that's what is required, how on earth are we going to deliver the medicine at this scale? How will we handle resistance? How do we deal with maintenance, not only physiologically, but in terms of cost? And ultimately, whether we should return back to paradigms of prevention. It's not that these are in exclusion of prevention, but to what extent we should return to paradigms of prevention and early treatment and early detection as we enter an arena which seems clearly very complex, and yet with a lot of promise for certain cancers. So I'll stop here. This was an attempt to give you a kind of landscape view of where some of the thoughts in cancer biology, cancer medicine are going, and open the floor to questions that you might have. Thank you very much.
So we now have time for a few questions. Yes.
Well, it was implicit, yes. Tell me. Yeah. So, what do I think of
this new effort?
Yes.
How is this going to work?
Well, so I'm going to answer the question on the condition that I get Lou to answer the question as well. So, because I think it's important to have I think it's obviously an important moment. We are, as many of you know, there's a moonshot campaign it's a little ill-defined, I think, at this stage, what moonshot exactly means, but many of the principles that I talked about are implicit in that moonshot campaign. So a deeper molecular understanding of cancer, including genetic and genomic annotation, followed by some kind of personalized therapy for individual cancers. That's at least implicit in the moonshot effort. It's a much larger effort, as you might know. So I think it'll end up with these questions. The questions that are in the moonshot effort are, in fact, these questions. How do we develop these therapies? How do we deliver the therapies? How do we manage the costs? What do we deal with maintenance? What do we deal with resistance? And what role prevention has? One thing that I'm optimistic about in this, and actually Lou and I were talking about this, is that one thing that may happen, and it would be very helpful if it happened, is that if we begin to collapse the silos of prevention, treatment, and cure. And by that I mean the following. When you detect an early cancer, could you, at that stage, find a molecular signature that would allow that particular cancer patient to be treated with some kind of adjuvant therapy, which would prevent the relapse of the cancer in the distal setting? So in other words, the idea of early detection now mishmashing or more effectively coordinating with a therapeutic attempt. Or, as people have pointed out, trying to use various chemicals as agents for prevention in particular high-risk groups. We've already had examples of that, for instance, the use of hormonal modulation in women at risk for breast cancer. So one thing that I think will maybe help is that if we take a personalized approach to cancer management, not only at the late phases of cancer, when there's so much tumor burden that in fact many of these attempts may fail in any case, but really move these personalized medical paradigms as early as possible to individual cancers. If that occurs, I think we may be able to manage some of the costs, we may be able to manage some of the, and get more effective treatments. For that to occur, actually, a much larger collaboration is needed because it will not be late-stage or end-stage cancer patients being treated with combination therapies. That will not work. What will have to happen is that early-stage cancer patients will have to be identified, or patients at risk will have to be identified and appropriately treated with the appropriate medicines for that. If that happens, I'm more optimistic. I'm less optimistic based on what we've seen so far at delivering at a scale level across the nation personalized therapies at this stage. I don't know, Lou, what your thoughts are.
Yeah, I agree with those comments. Just a bit of perspective, the total amount, it's not even clear yet what the total amount of money there will be for the moonshot, but almost certainly it will be far, far less than is needed. If it's $1 billion, that sounds like a lot of money, but most pharmaceutical companies spend $1 billion on cancer research every year. And so you're just adding to what other companies are already doing. Now, however, $1 billion spent in a way that forced us, for every patient, not just patients on clinical trials, but every patient who gets a therapy, particularly an immune or a targeted therapy, to share that information in a de-identified way in a centralized database across at least every cancer center, if not every hospital in the United States, would be immense. That would not cost anything other than just the cost of forcing people to share what they already are doing anyway. Obviously, the bioinformatics necessary to correlate these responses with mutational events across millions of patients instead of a clinical trial where you're looking at a few thousand patients is going to be difficult, but the opportunities to learn something from that are immense. So I think I was asked for my advice by the new NCI director, and that's the advice I gave him, and hopefully that advice gets pushed forward. Other questions?
Yes. In front and in the back.
Do you think there's a limit to how much we can affect the curve of chronic illness, given that people are living longer? And what role do you think that aging plays in this understanding?
So both important questions. So one thing is, of course, to be careful about the data to start with. So you have to age adjust the data. Otherwise, you begin to compare apples and oranges. So in order to figure out the real impact of chronic illnesses on mortality, the technically correct way to do it is to appropriately age adjust it, because as you're saying, the cohorts are aging, so therefore the weights have to be appropriately assigned. But the second question is, do I think there's natural limits? As you know, this is a major debate that's broken out more recently, whether humans with now left with chronic medical conditions are reaching a kind of age limit, as it were, or whether that age limit has been is real or artifactual. There was a big paper in Nature last week, I think, which made the argument that there's a plateauing of age. There was a reanalysis of that very same data, and that showed that, in fact, the plateauing was artifactual. So we really don't know whether there's any natural age limit to human beings dying of chronic diseases. What we do know, however, is that in chronic diseases, when interventions are successful, they can be very successful. And the great example, of course, is cardiac disease. In cardiac disease, the mortality of cardiac disease has been driven down dramatically over the last 10, 20 odd years, and then it has actually plateaued as well. But the dramatic decrease in cardiac disease using a combination of lifestyles, some impact of, among other things, cholesterol monitoring and decreasing agents, but really driven by lifestyle changes, has been dramatic. So we have examples in which chronic diseases have been altered. The epidemiology of chronic diseases has been altered during our own lifetime. The problem, as I showed with cancer, is, of course, that cancer is a unique chronic disease. It's a chronic disease unlike any chronic disease, partly driven by the genetic diversity and partly driven by the fact that we don't have effective medicines for many, many, many cancers right now. In the back. Yeah.
So there was just an editorial in the New England Journal of Medicine. It was in September. You got panic, basically questioning the whole paradigm of personalized cancer treatment, saying that more or less it's not really biologically feasible for all the reasons you've indicated. Too many mutations. The cancers are inevitably going to find resistance to whatever pathways we attack, and it's not really a worthwhile route to follow. I wonder if you have any thoughts.
Well, we have many thoughts on that. In fact, part of the presentation is a reaction to that idea that's been around. I personally sit in the middle. I think that the reality is that yes, this is a complex problem, as I demonstrated, but certainly in individual cancers where appropriate therapies have been directed, there have been landmark successes. We've shown some of those landmark successes. Cancer patients have benefited from those landmark successes. I think it is possible to develop personalized medicines against cancers, and that involves a combination of immunological therapies, metabolic therapies, lifestyle changes, and potentially other targeted therapies, stringing them together in the appropriate sequence. The question of whether they can be deployed at scale to make a dent in those large curves at a population level is a much more difficult question. I'm less optimistic about that, but I do not think that we should, certainly from my standpoint, are far from giving up the idea that these medicines can be deployed appropriately in appropriate groups of cancer patients. Yeah, again, love to have a comment on that.
Yeah, I think I'm always an optimist that we can do better with the tools that we have. We need better tools for sure. The advantage of targeted therapies over chemotherapy and at least therapeutic doses of radiation is you potentially preserve the immune system. And I think in the end, if we treated bacterial infections with antibiotics, that the first thing they did was kill all of your immune cells, and then they also killed some of the bacteria, we would always fail. The bacteria would develop resistance, and ultimately, unless the immune system was there to wipe up what was left, we would never be successful in treating bacterial infections. So I think we have to think about cancer in the same way. You've got to preserve the immune system or enhance the immune system with the same therapy that you attack the cancer. So the question is, we're not paying attention to that. How many people know what their targeted therapy does to their immune system? I think the second point is one that Sid made about early intervention being so critical. It's the same thing with bacterial infections. If a bacterial infection is too far along, you're not going to rescue the patient. The immune system is overwhelmed. The antibiotic, even IV, is not going to be sufficient. And that's when we do most of our clinical trials. We do our clinical trials in patients who've failed standard of care. That means they've been hit with 3 or 4 rounds of radiation chemotherapy, mutated the hell out of the cancer. Now you have hundreds and hundreds of mutations or thousands of mutations, more than were present when the disease was first detected. So detecting early, treat early with targeted therapies that spare the immune system, in my opinion, is where we have a chance to win. And the Herceptin example is good with that. You know, you saw the data on the trial. It was end-stage disease. Virtually nobody survived. And now that we have it as adjuvant therapy, I think the last numbers I heard are 95% survive, go long-term without relapse, 5 or 10 years. So that's, I think we have to learn from it. Gleevec is given as CML is still a stable disease. If in blast crisis, it doesn't work. And yet we're looking at blast crisis for most of the therapies we're giving. So we need to go earlier, and it's got to be more scientific, and we've got to pay attention to what's happening to the immune system. And so I'm the optimist that I'll agree the way we're doing it now, we're not seeing as much impact as we were hoping. But I think part of it is we're just not administering even the best drugs in the best way.
And certainly, I mean, ideas that were very hubristic maybe 5 or 10 years ago, which is, early detection for the sake of early detection, as you know, is useless. You can detect a tumor early. If you have nothing to do about that early detection, it makes no difference to a patient. Early detection with badly targeted therapies or inappropriate or inadequate or ineffective targeted therapies is similarly useless. So this whole paradigm will only work if you match the earliest possible detection of cancers with the appropriate molecular profiling, if you can achieve molecular profiling, which you can these days with very small amounts of material, and then use the appropriate molecular therapy to that. But now the question is imagining delivering that at scale across a huge, very obviously economically and geographically diverse population. Those are challenges that we haven't even faced yet, but we will face once these therapies become even marginally actionable. So I'm not a pessimist about personalized medicine. I disagree with the New England Journal editorial. In fact, there are plenty of examples in the cancer world, even with advanced cancers, such as multiple myeloma usually presents as symptomatic disease, that you can have partly targeted therapies. In multiple myeloma, they are not targeted against oncogenes generally. They're partly pathway targeted therapies, but they're successful in that disease, and they've turned around some of the mortality of the disease, not all of it, but have turned around some of the mortality of that disease.
I'll just make the one comment that probably all cancer therapies are targeted. It's just in many cases we don't know what the target is. So with that, Sid, thank you again for a fantastic lecture.